Okay. Now, the other examples for codominance is, one other example is the coat color of shorthorn cattle, wherein when it is homozygous dominant, then you have a red coat color. When you have a homozygous recessive, then you have a white coat color. But when both of them are present, then you have a codominant expression when you have a roan coat color, wherein it's a brownish red sort of a coat color. Now, like I was talking about codominance, it is not that one is being expressed and the other is not being expressed, or one is not being expressed completely as in incomplete dominance, but in codominance both of them are getting expressed. So if you look at the roan coat color of the shorthorn cattle, then you would see that there are hairs which are white and there are hairs which are red. So they are growing right next to each other. So if you have hairs, they are like growing right next to each other. Some of them are red, some of them are white in color, and therefore you have reddish sort of a coloration on the coat of the shorthorned cattle. And the same goes for the coat color of horses, where you have chestnut as the dominant, homozygous, white as the color when it is again homozygous recessive, but then you have a roan color which is again a brownish reddish color, wherein both the chestnut colored hair and the white colored hair are getting expressed, as in on the coat you would have chestnut colored hair right next to a white colored hair, and then a chestnut colored hair, and then a white colored hair. So from afar it would look like a reddish brown color, but if you inspect it right next to it, then you will see that there is a chestnut colored hair growing right next to that of a white colored hair. So the next thing that we will be studying about is multiple alleles. Now, unlike everything that we have studied thus far, everything, if you go for monohybrid cross or incomplete dominance or codominance, there is a gene which has two alleles. One is dominant, one is recessive. Sometimes both of them are dominant, or sometimes the dominant is not completely dominant, as in incomplete dominance. But multiple alleles are different in the sense that one gene is not going to have two alleles, but rather multiple alleles. So it will be more than two. And this occurs because a normal gene is undergoing mutation over a long period of time. So during the course of the growth of a population, so if you take into consideration the human population, as it is growing, there is going to be a gene that is going to undergo mutation as the population is growing. So that as the population is procreating and increasing in number, when the gene is getting passed on from parent to offspring, and then parent to offspring, and then parent to offspring, during this passage, that gene is going to undergo mutations, and that is going to give rise to not two but more than two alleles, and each of those alleles is going to have an alternate effect. So that is the thing with multiple alleles. It does not have two but more than two alleles, and it is a process which happens because of mutation over a period of time. Now, because it is multiple alleles, it is not something that can be observed in an individual. Like I was saying, an individual, one organism, a human being, for example, has two chromosomes of one set. So chromosome 9 will be one pair of the chromosome coming from the mother and one pair of the chromosome coming from the father. So each of those chromosomes is going to have two alleles for one gene, and therefore in an organism you can have only two alleles for a gene. So if you want to study multiple alleles, you will need to study an entire population, and only then you will come to know what other variants of that gene, of that allele are present in the population, and then you can figure out, okay, it is not just two of them which is found in one specific organism, but in another organism it's a different two allele, and in another organism a different two alleles which are found. Okay, and then because it is multiple alleles, it has more than two phenotypes. So it will not be just a dominant phenotype and then a recessive phenotype, but rather more than two phenotypes will be found in multiple alleles. Now, an example for multiple alleles, a very well-known example, would be ABO blood grouping in humans. Now, blood groups in humans are of 4 types, as you know, AB, and O, and they are separated as that because they have different antigens present on them. Antigens, or as they are known, isoagglutinogens. Now, they're nothing but a small chain of sugar which is attached to a protein on the surface of the RBC. So antigens, or isoagglutinogens as they are known, are nothing but a small sugar chain which is attached to a protein on the surface of the RBC. Now, there are 2 different antigens which are there, antigen A and antigen B. Now, both of them are coded by different genes, which is called as the gene I. It is termed as I because isoagglutinogens, and it is found on chromosome 9. Now, this gene I is going to code for an enzyme, galactosyl transferase. Okay, and galactosyl transferase does nothing but to transfer the sugar chain to the protein on the surface of the RBC, and that process is called as glycosylation. Okay, the process is known as glycosylation. Now, if the gene I is the gene IA, then it is coding for a galactosyl transferase, which is going to transfer galactosamine. If it is the gene IB, then it is coding for a galactosyl transferase, which is going to transfer galactose. Okay, so there are only 2 antigens, A and B, which are coded by IA and IB. If it is IA, then it is going to transfer galactosamine. If it is IB, it is going to transfer galactose. Okay, now there is a recessive gene which is I0 or IO, you can call it, which is going to code for a protein which is not going to transfer the sugar molecule onto the surface of the RBC. Okay, now if you have IA, then it is going to transfer galactosamine, and that person is going to have the blood group A. If it is going to be IB, it is going to be a galactosyl transferase which is going to transfer galactose, and that person is going to have the blood group B. Now, if both the genes IA and IB are getting expressed in a person, then both galactosamine and galactose are getting transferred onto the proteins on the surface of the RBC, and therefore that person is going to have the blood group AB. In case of blood group O, the person has the gene IO getting expressed. They don't have IA or IB, it's just IO. Then it is going to code for a protein, like I said before, which is not going to transfer the sugar molecule onto the protein on the surface of the RBC, and therefore they will have the blood group O. Okay, now in this example you can see multiple things. If it is just IA present, okay, so if you have the gene structure of IA, or IA, IO, then you have blood group A. Okay, and this is classical dominance which you would see in Mendelian genetics. Now, if this is going to get changed to B, so if you have IB or IB and IO, then the person is going to have blood group B. This is again classical Mendelian genetics where the dominant gene is going to get expressed over the recessive gene. Now, in case of AB blood grouping, you have both IA and IB genes. Both of them are getting expressed. So this is an example for codominance, which is going to happen here. And then you have double recessive classical Mendelian genetics again, in which case you have blood grouping O. Okay. Now, another example for multiple alleles is the peppered moth. The scientific name of it is Biston betullaria. Now, there are 3 different kinds of body color when it comes to peppered moth. One is the melanic, which is represented by M. The other is mottled insularia, which is represented by M dash, and then the typical one, which is represented by small letter n. Now, melanic is dominant over mottled insularia. Mottled insularia is dominant over typical. Okay, so melanic is going to be dominant over mottled insularia, and mottled insularia is dominant as compared to typical, which means that if there is a melanic gene present with a melanic gene, as in both the alleles are M in this case, or if one allele is melanic and the other is mottled insularia, or one of the alleles of the gene is melanic and the other is the typical, in all of these three cases, the expression of the gene would be only the melanic. So only the melanic allele would get expressed, because like I said, melanic is dominant over the other two. Okay, so if this combination of alleles are seen in an individual, then the body color would be black. All right, now the body color can be of mottled insularia type, which is a dark body, only if mottled insularia is present as a homozygous dominant allele, as in both the alleles are for mottled insularia, or one of the allele is for mottled insularia and the other is for typical. In this kind of allelic expression in an individual, then those moths would have a dark body. Now, a typical, which is a white-bodied moth, can only occur if both the alleles are a recessive allele, which is small letter m here. Okay, so only the white expression of the body color can happen only if it is a double recessive. And melanic is the most dominant as compared to the other two. This is another example for multiple alleles. So another example for multiple alleles would be the coat color in rabbits. The coat color in rabbit comes in 4 different kinds. The agouti, which is the wild type, the color would be black, which is represented by a capital C. Chinchilla, which is a silver gray color, which is represented by a capital C with CH. Himalayan, which is white colored with colored extremities, as in the entire fur is white in color, just the extremities as in the paws and the tail and the nose would be colored, which is represented by CH. And the albino, which is a complete white, a pure white, which is represented by a small letter C. Now, like in the peppered moth, the agouti is dominant over chinchilla. Chinchilla is dominant over the Himalayan. The Himalayan is dominant over the albino. So if you would take the genotypes which would give rise to agouti or chinchilla or Himalayan or albino, it would be of this kind. If you have homozygous double C, or if you have heterozygous CCH, or you have heterozygous CCH, or if you have heterozygous C and small letter c. All of these 4 genotypes are going to give rise to agouti. If it is chinchilla, then it needs to be a homozygous CH, or it can be a CCH, or it can be a CCH, small letter c. All these 3 would give rise to chinchilla. For Himalayan, again it will be a homozygous or a heterozygous with an albino allele. Both of these would give rise to Himalayan, and albino can only occur if it is a homozygous double recessive. Only if it is a small letter c allelic form, then it is going to give rise to a albino coat color. So this would be the different genotypes that you would need to have to get the 4 different colors. And like I said, agouti is dominant over chinchilla, and chinchilla over Himalayan, and Himalayan over the albino. Now these three are the three different examples which are there for multiple alleles, and like I was saying before, it is not something that you can see in one specific organism. For example, if you just take up this organism, you will not be able to know that there are different allelic forms which are there. Only when you study a population, a large number of organisms belonging to the same species, then you get to know that there are multiple alleles present for that one specific phenotype. And it is of course going to give different phenotypes, so you don't have two phenotypes like you would see in classical Mendelian genetics. You have multiple phenotypes like agouti or chinchilla or Himalayan or albi. The next topic that we will be talking about is dihybrid cross. Dihybrid cross in its principle is very similar to monohybrid cross, that you're going to take one character pure breeding plant with one specific phenotype in mind and then cross it with another pure breeding plant with the opposing phenotype. Okay, so the monohybrid cross, for example, was taking a pure breeding tall plant, crossing it with a pure breeding dwarf plant. Now with dihybrid cross, you would do the same thing, but like the name suggests, you are taking two phenotypes and not just one. So in this case, you would be taking round and yellow seed producing plant and cross it with a wrinkled and green seed producing plant. Now if you go back to the previous talks, I was talking about round being dominant over wrinkled and yellow color being dominant over green. So the genotype for these parents, because they are pure breeding, would be homozygous dominant. So they are round and yellow, which is being crossed with wrinkled, so small letter r and small letter y. Okay, so these are the parent generation. The gametes that you would get would be r and y being crossed with small letter r and y. Now this is one thing that you would need to keep in mind when you are putting down gametes, that you are not taking gametes for single alleles, but rather you are taking both the alleles as one single gamete. So it will be R and Y, R and Y. Same goes for R and Y and R and Y. So here you would have two gametes being formed. But because they are the same, I'm not going to write it again and again. Now if you're going to cross it, like in monohybrid cross, draw a Punnett square. So you would have capital RY, capital R, capital Y, small letter r, small letter y, small letter r, small letter y, this being the male, this being the female. Crossing it, you get capital R, capital Y, small y. Same goes in all of them. Like in any F1 generation, you are not going to get different phenotypes in the F1 generation. The phenotype is going to be the same. The genotype is also going to be the same. Just the difference being, in the parent generation, you have homozygous genotypes. In the F1 generation, you have heterozygous genotype, which being capital R, small letter r, capital Y, small letter y. This is for the F1 generation. This is the heterozygous genotype. The phenotype being round and yellow, because of course round is dominant over wrinkled and yellow is dominant over the green. Now if you're going to self cross F1 generation, the F2 generation would be crossing capital R, capital Y, small y with itself. Moving on with dihybrid cross, I was talking about selfing the F1 generation. So from the F1 generation, you get a heterozygous plant, but it is going to have the phenotypes which are dominant because you have both the dominant genes, right? So you're going to have round and yellow colored seeds. Now if you're going to self cross them, you would get the gametes capital R, capital Y. So capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So for each parent, you're going to have 4 different gametes. Now, if you're going to put up a Punnett square and cross all the 4 different gametes with each other, you're going to get this result. Okay. Now, the phenotype is going to come out in the 9:3:3:1 ratio, where 9 being the dominant, meaning you have round and yellow. Then you have 3 of them, which would be round and green. So you have round and green here, round and green here, and round and green here. So 3 of them being round and green, 3 of them being wrinkled and yellow. So wrinkled and yellow, wrinkled and yellow, wrinkled and yellow, and 1 being the double recessive, which would be wrinkled and green here. The rest of them are all, the 9 of them, which will be the dominant, which is round and yellow. So phenotypically, if you go for F1 generation to F2 generation in a monohybrid cross, you have a 3:1 ratio phenotypically. But here it's a 16 square Punnett square, and in that you get a phenotypic ratio of 9:3:3:1. Now that ends the dihybrid cross. Now the Mendelian laws which were there based on the monohybrid cross apply for the dihybrid cross also. So you have segregation of the gametes, unity of gametes, and the 3 laws which are there, they are applied for both the monohybrid cross and for the dihybrid cross. That is why when you take the parent, you're going to differentiate the gametes, because each of them is one unit. Okay, the next thing to know would be the dihybrid test cross, and like in the monohybrid test cross, you're going to cross the F1 generation with the double recessive parent. So you're crossing the F1 generation, which is a heterozygous plant, with a double recessive parent, which will be wrinkled and green colored seeds being crossed with the round and yellow colored seed bearing plant. Like before, when I was talking about dihybrid cross, the segregation of gametes will occur, and so you have 4 different gametes coming from the F1 plant, which will be capital R, capital Y, same, capital R, small y, small r, capital Y, small r, and small y. From the double recessive parent, you will have 2 gametes coming, but because they're repetitive, we are going to take only 1 into account. So if you're going to get gametes from here, it would be small r, small y, small r, small y. So here there will be 2 of them, small r, small y, and small r, small y, but because they are the same, it is repetitive, so we are going to take into consideration only 1 gamete from the double recessive parent. You draw a Punnett square again, put in all of the gametes from the F1 progeny and the double recessive parent. You have this as the genotype of the plants that you would be getting. Now they are in the 1:1:1:1 ratio, like you would find in the test cross when you would be doing test cross in monohybrid cross. The progeny will always be in 1:1 ratio phenotypically and genotypically also. So here you have a round and yellow, then you have round and green, wrinkled and yellow, and wrinkled and green, all of them being in the 1:1 ratio, and that is a dihybrid test cross. It is exactly the same as you would be doing in a monohybrid test cross. The only difference being here you are taking 2 characters. In case of monohybrid test cross, it would be just 1 character which should be taken into account. Going away from Mendelian genetics, we have something called as complementary genes. Complementary genes are when there are 2 or more non-allelic genes which are going to interact with each other to give rise to 1 phenotype. Okay, so it is not the same allelic genes, meaning that if you take the alleles for tallness, for example, you have the allele capital T and the allele small t. Now these two alleles belong to the same gene. Okay, but when you have non-allelic genes, as in if you take the genes for, let's say, tallness, and then you take the genes for example, the shape of the seed in case of garden pea, which would be round and wrinkled. So these two are alleles for the round and wrinkled, so the shape of the seed, and these two are the alleles for the plant height. Now they are not related to each other, so these are two different alleles, but they are going to, if they are going to, they don't in this case, I'm just giving an example, if they are going to interact with each other and give rise to one specific phenotype, then that is called as a complementary gene. So when you have two different non-allelic genes interacting with each other to give rise to one phenotype, then they are called as complementary genes. Now complementary genes, like I said, produce one phenotype, and they need not be found on the same chromosome, meaning both the genes, so two different alleles of two different genes, so 4 alleles in total, all of them need not be found on the same chromosome. They can be found on the same chromosome, but even if they are not found on the same chromosome, they are still going to interact with each other and give rise to one phenotype. And another condition with complementary genes is that both of the genes need to be in a dominant state. So both the genes, as in both the alleles, so at least one of the allele of two genes need to be in a dominant state, and only then it is going to give rise to the phenotype. So if I go back to the example that I was giving before for height and the shape of the seed, one needs to be the dominant allele and the other can be a recessive allele, or it can be a dominant allele. It does not matter, but one needs to be a dominant allele, and the same goes for this also. So at least one of them needs to be a dominant allele, or two of them can also be dominant, it does not matter, but at least one needs to be a dominant allele, and only then the phenotype will get expressed. Okay, an example for complementary genes is the flower color in sweet pea. Now sweet pea is different from garden pea, so it's not Pisum sativum, but rather it's Lathyrus odoratus. Okay, now the flower color has a complementarity in it. So there are two flower colors in the sweet pea. It's either white or purple in color. Now white colored flowers are found when the complementary genes are not complementing each other, as in neither of the alleles in neither of the genes are dominant. Okay, so like I was telling, with complementary genes, at least one allele in both of the genes need to be dominant. If it is not, then it is not going to show the phenotype. So when the phenotype is not being shown, as in when the genes are not complementing each other, then the color that you would find would be white. So if you take a pure breeding white and cross it with another pure breeding white, then you have in the F1 generation a purple color, because the non-dominant one from one and the non-dominant one from the other are going to complement each other, and you would get a double dominant gene interaction, and therefore you have a purple color in the F1 generation. Now if you're going to self-cross F1 generation, in F2 generation, unlike the 16 Punnett square which you would find in dihybrid cross where you have 9:3:3:1, you have a different ratio here. You would have a 9:7 ratio, because you're getting only two phenotypes. You're either getting purple or you're getting white. So whenever both the genes are not dominant, so let's say that C represents white and P represents purple. Okay, so C is the gene for white color and P is the color for the gene for purple color, let's say. So if both the genes are not dominant, so you have two genes C and P. Now one is dominant here, so you have two dominant genes but two recessive genes in case of the purple, so it's not going to give purple color, it's going to give white color. If you go the other way, one dominant, one recessive, so one gene is still dominant, but the other is still double recessive, so you're going to again have white color. Here you have a double recessive but a double dominant, so again you're going to have a white color. Double recessive, here heterozygous dominant, but still it's going to give a white color. Now here both of them are double recessive, so again you would have white color. So these are the these are the different phenotypes that you will come across if you're going to have white colored flowers. So if both of them are dominant, so if you have a heterozygous dominant, you will get purple. If you have homozygous dominant, you would have purple color. So it needs to be either heterozygous or homozygous dominant, only then you have purple colored flowering plants. If it is a kind of genotype where only one of the gene is in a homozygous or heterozygous dominant type, and the other gene is either homozygous or heterozygous recessive, then you're not going to have the purple color, the phenotype which needs to be shown, but rather you would have the recessive phenotype, which is the white. Okay, another example for complementary genes is the deaf mutism in human beings. If you take a deaf mute person and mate them with another deaf mute person, so this is a male being mated with a female, you would have normal human beings, as in they have normal hearing and normal speech pattern. They are neither deaf nor mute. Now if you would self-cross F1, now because we are talking about human beings, we cannot self-cross them because interbreeding is not allowed in human beings. So for F2 generation, we can do pedigree analysis. So if you're taking a normal human being from the F1 generation and crossing with another normal human being from the F1 generation, so a selfing is happening. So let's, for example, think that the two genes which need to interact with each other to give normalcy is A and B. So randomly thinking that A and B are the genes which need to interact with each other to be able to have normalcy. So if normalcy is found, then both A and B are in dominant condition. So either they are homozygous dominant or heterozygous dominant, but because both of them have either at least one dominant allele, they are going to interact with each other and give rise to a normal human being who is not deaf nor mute. But if that is not present, in which case there is one allele which is dominant but the other is not, the same example like that is found in the sweet pea flower color. So if both of them are not dominant, so here we have a homozygous dominant gene and a homozygous recessive gene. So this is going to give rise to a deaf mute person. You have a homozygous, heterozygous dominant and a homozygous recessive, again going to give rise to a deaf mute person. Homozygous recessive but homozygous dominant in the other, B gene, again deaf mute. Homozygous recessive but heterozygous dominant, still going to give rise to a deaf mute person. Homozygous recessive in both the genes, all of them are going to give rise to a deaf mute person, but only if it is either homozygous dominant, as in you have this genotype, or you have heterozygous dominant, which would be this genotype. Then you have normal human beings who would have normal hearing and normal speech pattern.